Hello everyone. We're gonna start this video by talking about biofilms. As we said before in the classroom, that biofilms are thin layers that contain microorganisms, many type of microorganisms, actually any type of microorganisms. So biofilms, because the prefix bio indicates microorganisms or living things, so they might contain bacteria, they might contain viruses, they might contain fungi, protozoa. So they're all microscopic organisms that we cannot see with the naked eye. And as we said before, in sciences, films or film means a thin layer. So it's actually a thin layer of microorganisms, and these microorganisms should be secreting a sticky substance, a slimy sticky substance, or a gluey substance that helps them stick to each other and to the surfaces. In addition to this, of course, this thin layer of microorganisms, in addition to being sticky, it should be moist, meaning that it should contain a certain amount of water, however small it is, because all these living organisms, as you know, require water. Biofilms can form on any fomite, and fomites are solid substances on which biofilms can form, and therefore fomites are an important method of or an important vehicle of transmitting infection from one person to another. The importance of biofilms is that they can form on any surface. So whatever the type of the substance that forms the surface is. So they can stick to metals, like in doorknobs, for example. They can stick to plastics, like in cutting boards that we use to cut food. They can form on the floors, on soil particles, on biological tissues. Of course, you might have a biofilm on your skin if you don't wash frequently. And most importantly, and this is what we're going to discuss now, is that they can form on medical implants. What are medical implants? As their name indicates, they are something related to medicine that we use and that we implant on the surface of the body or inside the body, and they might be tissues, living tissues, or they might be devices. So medical implants are tissues or devices that are placed either inside the body or onto the surface of the body. Just like biofilms can form on different types of materials, implants can be made from different types of materials. So in skin grafts, for example, when we use a skin graft to replace a burnt area of the skin, this is a type of implant that's made of living tissue. As we said before, we might use a bone graft when you have two ends of a long bone, for example, that are far away from each other and whose healing is almost impossible because they are far away from each other. So we put the bone graft in the middle in order to help joining them. And then sometimes we join them with something made of metal, of non-reactive metal, as we said before. And that non-reactive, it's important for the metal to be non-reactive. You wouldn't add, for example, screws and a plate to fix the bone, and you wouldn't be expecting the plate to be oxidized, for example, to be reactive inside the body. So we have medical implants that are made of bone. Sometimes we have medical implants that are made of plastic, like the chemotherapy port that we're gonna discuss on the next slide, or they might be made of ceramic, like for example the caps that we put on some teeth. We sometimes make them out of ceramic. What are the functions of what are the functions of all these medical implants. So this last paragraph will be talking about the functions of the implants. So the first important function of the implant is to replace a missing body part. For example, as we said before, I'm just reviewing what we said in class. If you have an amputated limb, like when, for example, a certain area of the world is exposed to wars, and due to explosions and bombs and so on, people might have injured limbs, limbs that are exposed to severe infection, or limbs that are severely damaged. And in such a case, in order to save the life of the victim, then we amputate the limb. We remove that part of the limb. So people require a replacement, and that replacement is what we call a prosthetic limb. So prostheses are nothing but medical implants that are used to replace a missing part. So every time you start by it is a medical implant that is used for. So as you see on the next slide here, this is an example of a prosthetic limb that can be used as an implant, in a medical implant, and it's joined to the thigh of the person who had an amputated limb. Okay, so first of all, it's prosthetic, meaning that, or a prosthesis is an implant that replaces a body part. But we also have implants that control or monitor body functions. Monitor means control body functions. And the best example for this is what we called the artificial pacemaker. The artificial pacemaker. The artificial pacemaker, or the pacemaker in general, as an implant, it is an implant. Again, I start by it is an implant that controls the heart rate. Of course, we do not jump to using the artificial pacemaker from the very beginning, but we should start first by analyzing the causes of irregular or fast or slow heartbeats. For example, if the patient does not have adequate sleep, if the patient drinks a lot of coffee or a lot of tea, or if the patient is too anxious and stressed for a reason or another. So we first remove the underlying reasons, and then we give medication. But if the medication is not enough to relieve the palpitation, then we will fix an artificial pacemaker. And by the way, talking about palpitations in the heart mean one of three things. So it's either a too fast heart rate, or a too slow heart rate, or a too slow heart rate, or it might be an irregular heart rate, or it might be an irregular heart rate. How do we fix the pacemaker? Here you have the heart, and we insert it under the skin, and there is a small plastic tube that passes through the veins until it reaches this area, and through this tube we can pass electric charge to the heart to control the heart rate. Originally, in normal persons, in the right atrium of the heart, there is an area of the muscles of the heart that is called the SA node, and that acts as the natural pacemaker of the heart. So we don't use the artificial pacemaker. We don't implant the artificial pacemaker of the heart unless we have palpitation. And if we have palpitation that is intractable means that we cannot treat by medication or by removal of the underlying factors. This means simply that this natural pacemaker of the heart, the SA node, is diseased, and that we require an artificial pacemaker. the third function is to deliver medication. So number one, to replace a missing body part. Number two is to monitor body functions. And number three is to deliver medication. As I told you, in the classroom, sometimes we use something called the chemotherapy port. Of course, you know that chemotherapy are medications that are usually given intravenously through the veins. And we deliver that kind of medication to kill the cancer cells. And sometimes we need a device, an implant. We need to fix this implant to implant it under the skin because it will act as a port. And port here means a tool to deliver the chemotherapy medication. As you see here, we make a small incision and we implant the chemotherapy port, the pump here under the skin, and you have palpation points. Those two palpation points help you be guided where to insert your needle, and then the chemotherapy medication is delivered through the catheter that is inserted in the vein. Why are we doing this? Because as we said before, when we when we give the intravenous medication using a syringe, for example, directly intravenously, this means that we might suffer from the complications of direct intravenous injection of the chemotherapy. And the first complication is blood clotting at the area, which would completely occlude the blood vessel and prevent the flow of the chemotherapy medication to the whole body of the patient. So the first problem is clotting and obstruction. And the second problem from direct intravenous chemotherapy is extravasation. And when we say extravasation, we mean that there is leakage of the chemotherapy outside the vein. And when the chemotherapy leaks outside the vein, there is a severe burning sensation and pain that the patient cannot tolerate. So in order to avoid those two complications of the direct intravenous injection of chemotherapy, we use the chemotherapy port, and it will be fixed there until the patient finishes all the sessions of chemotherapy that might last for 2 months or more. So we use this whenever people need like repeated, frequent, long-term injections of chemotherapy. And this was the third application. The fourth application, of course, this is another application that's not written in the text, but that is related to the word prosthesis. So here, as you can see, this is the femur bone, and the femur bone originally has a head, and that head is made of bone. But if there is damage to the joint, which we call osteoarthritis, severe damage to the joint, the hip joint, I mean, then, or there is a fracture, for example, that does not heal, then we can put an artificial joint. This type of surgery that we use in severe advanced osteoarthritis or severe damage of the joint. This operation or surgery where we replace the original joint by a metallic joint, as you can see in the picture, this is what we call total replacement of the joint. If you do this in the hip joint, then it would be called total hip replacement. If you do this in the knee joint, then it would be called total knee replacement. And these are the two famous types of orthopedic surgery that where we do total replacement of the joint. There remains one important application, which is providing support to organs and tissues. We've mentioned one type of support, which is fixation of the bones using the plate and screws, to fix the joints. And we talked about this a couple of minutes ago, but supporting organs and tissues can be also through another application, not only plates and screws in bones, but through through stents in the coronary arteries. What are the coronary arteries? The coronary arteries are the arteries that supply blood to the cardiac muscles, and sometimes they get obliterated or obstructed by fat. So we open them by catheterization, and we keep them open through a stent. So we first do catheterization, which we call in Arabic astara or qastara, catheterization, and then we fix a stent, which we call in Arabic du'ama, which means support. So here, as you can see, we have this is a section in one of the branches of the coronary arteries. And as I said before, the coronary arteries are the arteries that supply oxygen to the cardiac muscles. Of course, you know that when the heart pumps blood, when the heart muscles contract, they pump blood loaded with oxygen and glucose to all the tissues of the body. But in order for the myocardium of the heart or the cardiac muscles to be able to do this, they need themselves to get energy, and this is through the coronary arteries that branch on the outer surface of the muscles of the heart, and that are very small and very liable to be obliterated. And as you can see here, you can see a plaque, and that plaque is called an atheroma. An atheroma is a plaque or a deposit of fat. And this deposit of fat is very harmful because it does two harmful things. The first thing is that it renders the diameter of the blood vessel smaller, so you would have a narrow coronary artery, and a narrow coronary artery means less blood flow to the muscles of the heart, which is, of course, as you can guess, injurious. And the second thing is that the deposition of fat in the inner lining of the coronary arteries is irregular. It looks like the scales of a fish, so it's very irregular, so it renders the inner wall of the coronary artery rough and stiff, and this favors the occurrence of clotting and occlusion of the coronary arteries. And of course, you can anticipate what dangers can occur if we have the coronary arteries rough, stiff, liable for clotting. So what we do is catheterization and stenting. Catheterization means that we will pass this catheter inside the coronary arteries, and that catheter carries a deflated balloon around it. We cannot see it, but if this is the catheter, so we have a deflated balloon around it. And then here, as you can see, this white shadow I am pointing at or going over is the balloon when it is inflated. So when you use the catheter, you use it to guide the balloon towards the coronary artery. And then when we use the balloon, you use it to inflate it. And when you inflate it, you increase the diameter of the coronary artery. You make the coronary artery wider. And then at the end of the day, if you leave it like this, it will go narrow again. So you have to fix this stent that you can see, this lattice that you can see, lattice or network, or we call it, the medical term is stent, and you keep that stent in place. Sometimes the stent itself for releases medication for a few months, and those medications are dilators of the coronary arteries to increase the diameter. A few students ask me about the catheter. First of all, where do we introduce the catheter? At first, we introduce the catheter if this is the body of the patient, like this, and we will make a small incision here, and that small incision will allow us to intrude the catheter into a major vein here, and then through a bigger vein until we reach the heart. All this is monitored on a screen. You cannot do this without monitoring on a screen. By having said this, we would have finished everything about biofilms and their medical applications. The only remaining detail that I would like to say is that we sometimes remove the implant, or we keep it in place forever according to the circumstances. Another very important example of biofilms is the biofilm that forms on the cutting boards. This is a scanning electron microscopic picture, which is colored, of course, for the cutting board. So why electron microscope? EM refers to the electron microscope because the electron microscope not only allows you to see bacteria just like the light microscope, but it also allows you to see smaller microorganisms like viruses and to see all the details of these microorganisms. This proves that however apparently clean the cutting board is, yet there are plenty of microorganisms on it. What does this imply? What does this mean? This, first of all, means that we have to frequently change the cutting board. You don't, especially when they are made of plastic, if they're made of metal, which is unusual because of the scratching that occurs on it from the knives. So you should frequently change it in order to minimize the amount of colonies that might form on top of the cutting board. But the second important thing, other than frequently change, is to use separate cutting boards, meaning that if you have a cutting board for meat and chicken, then don't use this cutting board for vegetables and fruits or for cutting bread and cheese, because of course raw meat and raw chicken can contain microorganisms that are sometimes serious and highly infectious. Sometimes on boats or inside drainage systems, especially if they are small drainage systems with very shallow water inside, sometimes we have biofilms that are made of algae, and those biofilms are usually very slippery. And the best example is the example of the rocks that form of the biofilms made of algae that form on top of the rocks besides the sea, and that are very slippery and very dangerous to stand on. And now we're going to talk about the classification of the different types of infection. And of course, just like in any type of classification, we have different methods of classification of infections. So we can, the first criterion to classify infections is the presence or absence of symptoms. The second criterion is about the extent of the infection in the human body. Of course, if you harbor microorganisms, whether this microorganism is a friendly flora or a pathogen, then and you still don't have symptoms, then this is called subclinical infections. Clinical means with apparent symptoms. So subclinical means without apparent symptoms. You might understand how you might be harboring a friendly flora with no pathogen. You might also understand how you might be harboring a potential pathogen like Staphylococcus aureus, which is, of course, essentially a friendly flora without showing symptoms. But you might not imagine how you might harbor a more serious thing, which is an actual pathogen, for example, a virus, because when it comes to viruses, we do not have friendly viruses. So an actual pathogen like a virus, you might be wondering how I might be harboring in my body a virus without showing symptoms. Of course, we mentioned this before, and we said that in almost all viruses, there is a stage called viremia, when the virus is circulating inside the blood, but not yet having reached its target organ, because every virus has one or more target organs. For example, COVID-19 principally had the respiratory system as a target organ. So viremia is a stage when you have the virus circulating in the blood, having not yet reached its target organ and therefore not replicating. And therefore, since the virus in viremia, in the stage of viremia, is not replicating, then it does not give symptoms. And then once the virus reaches the target organ, it is, as you know, an obligate intracellular parasite. It cannot reproduce unless it is present inside a living cell. So it starts replication. And when it starts replication, it starts giving symptoms like fever, body ache, and so on. This stage of viremia is a stage of subclinical infection. So this stage of viremia, first of all, represents a good example for subclinical infection. Furthermore, this stage of viremia, as you know before, or the patient in such a case is called a carrier. And of course, you might be infectious during that stage without the community around you knowing that you are infectious. And that period of viremia is called incubation period. So what is the incubation period? The incubation period is the period of time during which the microbe or the pathogen is circulating inside your body without showing symptoms. On the other hand, once the symptoms appear, then we are dealing with a clinical infection. The second, in addition to dividing infection into clinical and subclinical infections, we can divide infections according to another criterion, which is the extent of infection in the human body, how spread the infection is in the human body. So if the infection is located at the site of entry of the microbe, like this very small boil or abscess, and an abscess, as you know, is a collection of pus or dead white blood cells that is surrounded by a wall. So if you have a collection of pus surrounded by a wall, then we call it an abscess. So an abscess is a very good example of local infections, because all the symptoms of infections that you can see, symptoms and signs, of course. Let's talk here about the signs that we can see in the picture. We can see redness, we can see swelling here, and we can see the appearance of pus. All these signs are located at a very small area, which definitely represents the site of penetration of the organism. But if the symptoms are widespread in the body, involving many body systems, and giving widespread symptoms and signs because they are affecting many organs inside your body, then this would be systemic infection. What's the clinical picture of local infection? So here we are dealing then with the signs and symptoms of local infection. The signs and symptoms of local infection include most of them, in case of local infection. For example, you have a wound here, and then the bacterium entered and made a boil or an abscess, as we saw on the previous picture. So you start by having a small wound or even a minor scratch in the body. And this simply means that you have tissue injury. And if there is tissue injury, there is always release of histamine. And histamine is considered a chemical mediator of inflammation. It is a chemical mediator of inflammation. What does this word mean, or this term mean? Mediator means facilitator of inflammation because it causes inflammation, meaning that all the signs of inflammation that you suffer from result from histamine. How does histamine do all this? Another question is, how does histamine result in all these symptoms and signs? Histamine will cause vasodilatation. It will cause widening of the blood capillaries, dilatation. And if it causes widening of the blood capillaries, then there would be more flow of blood to the area. More flow of blood to the area will mean that this area will be red instead of being anything else, pink or whatever. This area will be warm because blood comes from the core of the body and is warm. And this means swelling because more blood to the tissues means more liquid and more leakage of fluid outside the cell. But it also means local pain because histamine stimulates the pain nerve endings. So all the symptoms you see, heat at the site differs totally from fever. Heat at the site is the local warmth you find or you feel in the swollen red areas, and you have pain, local pain, you have local redness and swelling. All these are nothing but the cardinal signs of inflammation. These are the cardinal signs of inflammation. Of course, the appearance of pus is late because pus is nothing but dead white blood cells. Sorry. So if you have dead white blood cells, then you have pus. And why do we have white blood cells in the area? Because they were recruited to fight the microbe. And after having fought the microbe, they die and they form pus. If you have a big amount of pus, then you have a very bad smell that will be smelt only if the abscess ruptures and pus is released. And this is the odor of not only the dead white blood cells, but which are essentially the main component of pus, but you will also have dead tissues, the injured tissues, and you will also have dead bacteria or dead microbes. All this shares in adding more foul odor to the area. Systemic fever, meaning that the whole body suffers from elevation of the core body temperature. And when I say core body temperature, I mean the temperature of the inside of the body as measured by a thermometer. This is totally different from heat at the site. But let's agree that systemic fever is very rare in case of local infections. We said before that there are factors that help the infection to be localized. First of all, the dose of the microbe. If you have a small amount of microbe that entered through the wound, then you will have mild symptoms. The second is how virulent the microbe is, how powerful the microbe is the microbe too virulent, and therefore it can overcome all the barriers, all the immunological barriers, or whether the microbe is weak and easily destroyed by the immune system. And the third factor is how effective your immune system is. How effective your immune system is, okay? And then we move on to the systemic effects of infection. Of course, the first is body aches. All the systemic infections of infection result from the microbe circulating in the blood, but not only circulating in the blood, the microbe releasing harmful chemicals into the blood. The microbe releasing harmful chemicals into the blood, and this helps occurrence of these systemic or generalized system. So you might have headache only, or you might have generalized body aches. The second symptom is chills. And if you find somebody chilling, then this means that he might be feverish. Of course, we have a fever because these harmful substances that are released from the microbes and that are called pyrogens, these pyrogens go to the brain and reset the temperature regulating center in the brain to a higher level, meaning that inside the brain there is a certain heat or temperature regulating center, and that temperature regulating center always regulates the body temperature at 37 because this is the most favorable to enzymes. So if you reset, if these pyrogens that come from the microbes reset the heat regulating center of the brain to a higher temperature, say for example 39, of course this would affect all your body enzymes. This will diminish the function of all your body enzymes, and that's why you feel fatigued and you always want to sleep when you are feverish. Furthermore, these chemicals might trigger the vomiting center in the brain and give you a sensation of nausea or even vomiting. Weakness, of course, is again due to this load of toxic material that is released in the body. And now we're going to talk about something we discussed before several times, but we're just wrapping up: infection transmission. Infection transmission, of course, as you can see, can occur from one person to another, or it can occur through again. So I think that the numbering is not correct. So first of all, let's see the broad titles. From person to person, this might be through touch. So when we say from person to person, we mean either directly or indirectly. So first of all, we have touch, just like we talked before about the nasal carriers, and when they sneeze or cough, and droplets of saliva can either flow through air or through dust, but here we are talking about touch, which is a direct person-to-person transmission. So touch is direct person-to-person transmission, meaning that you cough or sneeze into your hands, and then you have just wiped your nose off and you shake hands with another person. So you might transfer a potential pathogen like staphylococcus, or you might transfer an actual influenza virus like the rhinovirus. But there is another way of transmitting infection from person to person. It is through contaminated body fluids. And through contamination of body fluid or under contaminated body fluids, we study contaminated blood and contaminated saliva. In such a case, there will be no, it's again person-to-person infection, but in such a case, there wouldn't be a direct way of transmission of infection, but an indirect way. Saying, for example, that you're giving blood for blood transfusion, and of course, this shouldn't occur because we should examine the blood that is donated for transfusion. But suppose that it was an emergency transfusion and the blood contains hepatitis B virus or HIV virus, which is human immunodeficiency virus that results in AIDS. And of course, you know that the hepatitis B virus results in infective hepatitis or viral hepatitis. In such a case, it might be through a blood transfusion. You are receiving a blood transfusion that contains the virus, or through sharing used needles. Of course, this is something that should be completely banned, but anyways, if it happens, like in drug addicts, for example, the syringe, the previously used syringe might contain traces of blood from the previous person. Another indirect way of transmitting infection through body fluids is through saliva, because saliva is a body fluid, and this is what we used to call droplet infection. So you might inhale droplets in air, you might inhale directly droplets that are coming to you from air, even if you're not standing in front or talking to that person directly, but the droplets might reach your place or the point where you are standing and you inhale them, or you might inhale the droplets directly, or you might inhale dried out dust particles, as we know that that contain the droplets. So we have airborne infection and foodborne infection that are also related to droplet infection. I think we need to redo the numbering and the lettering of those items, because droplet infection and dustborne infection are examples of airborne infections. So what we discussed here under droplet infection, that is directly transferred to another person, this is airborne infection. But if it dries out on dust, this would be dust-borne infection. So air can carry dust. Here we are putting them together under airborne infection, because as you know, whenever it comes to classification, there are different ways of classifying and numbering the items, because you can classify from different perspectives. So the classification here is based on the fact that whether you are transferring saliva droplets directly, or you are transferring dried out saliva in dust particles, anyway, both will be carried by air. So some literatures or some references separate airborne infection from dust-borne infection, and others put all types of droplet infections under airborne infections, because they are, after all, whether direct droplets or dust particles, they are carried by air. So you can choose whatever classification suits you, but at the end of the day, you have to say everything correctly and justify it. Foodborne infection is considered one type of droplet infection, of course, because if you eat food that is contaminated by droplets of saliva, then it might like infect your gut. And in such a case, it would be mostly a gastrointestinal disease. So you might have gastroenteritis, which is in Arabic nazla ma'awiyah, or food poisoning. Of course, gastroenteritis is a moderate form of food poisoning. In gastroenteritis, the most important characteristics that you have inflammation of the stomach, gastritis, leading to vomiting, and inflammation of the intestines leading to diarrhea. So vomiting and diarrhea are the characteristic symptoms of gastroenteritis. Furthermore, in food poisoning, you still have vomiting and diarrhea, but that are more pronounced, because in case of food poisoning, the bacteria are secreting severe toxins in the blood, and those severe toxins or severely harmful toxins are transmitted via blood or transported via blood to the stomach and the small intestines, resulting in vomiting and diarrhea. Of course, these are a few examples of how droplets can cause foodborne infection, whether that the food handler doesn't have clean hands and is not wearing gloves, or that the food handler has sneezed or coughed onto the dishes or the food types he is or they are handling, or that the kitchen utensils themselves are infected. Another way of transmitting infection is food poisoning, as we said before, and food poisoning here, why are we putting it as a separate category? Because we are talking mainly about a type of microorganisms like salmonella that is essentially present in raw meat. But these are not worms. These are microbes. So Salmonella is a bacterium that causes food poisoning, and that is essentially transmitted via polluted raw meat, and E. coli is essentially transmitted via polluted fresh water, and they both cause food poisoning. And here we are talking about the role of some vectors like the houseflies. Here, a vector is a living organism that is responsible for, or that results in, transfer of infection from the source to the human body. Why am I saying the source? Because the source might just be fecal waste that come from an infected organism, and that contains salmonella and or E. coli. And when the housefly stands on this infected food, then you know that the legs of the housefly are hairy, and their hairs usually, not accidentally, it's accidental, but it's very common that the hairs in the legs of the housefly carry very small particles of fecal waste, and when they stop on another or stand on another food item, then they simply transfer these substances. We also have fomites or fomite-borne infection, and fomite-borne infection is fomites are non-living objects, as you know, like doorknobs, like kid toys, like towels, like toilet seats, like barbed wires, like beddings that can carry disease-causing organisms. That's why if you exchange towels during, for example, exercising, then these towels might transfer a very famous fungus that we talked about before, which is the trichophyton, and that fungus, trichophyton, causes athlete's foot. We talked about athlete's foot before, and we said that it is a fungal infection that is transmitted via fomites, and that prefers, of course, the damp, warm, like dark, moist areas of the body. And amongst these areas, we have the areas in between the toes. It causes inflammation and, of course, severe itching and whitish plaques. And the skin in between the toes, as you know, as we said previously, might be sored or fissured, and it of course needs an antifungal treatment. So fomite-borne infection was the last mode of transmission of infection. And then we had, I think we jumped a slide. Yes, because fomite-borne infection is number 7. And then let's go retrograde to classify things correctly. So fomite-borne infection is number 7, and we studied trichophyton, we studied athlete's foot, and athlete's foot is Tinea pedis, athlete's foot without an A, because with an A, this is another thing. So it is T-E-N-I-A, Tinea pedis. This is the scientific name for athlete's foot. And then before fomite-borne infection, we had number 6, which we are going to explain now. And this is the last slide to explain, is insect-borne infection. And then we have number 5, waterborne infection. Number 4, foodborne infection. Number 3, airborne infection. Number 2, contaminated blood or other body fluids. And number 1, touch. I think that the best thing to correct the system of classification is just to erase or delete this title. And if we do this, all the other numbers would make sense. What did we skip? We skipped the waterborne infection. Of course, we have talked about this while talking about the digestive system. If you drink polluted water, and that polluted water is polluted with animal feces, just like we said in the case of food poisoning, then you might acquire typhoid or cholera. Cholera is just like food poisoning, severe vomiting and diarrhea, but it's even more more severe than food poisoning, because not only does it cause vomiting, but also watery diarrhea. Watery diarrhea is a severe type of diarrhea where there is frequent defecation of a very liquid stools, just like water. And of course, if it is not with severe fever, of course, and body pain, if it is not treated, it would, or it might lead to death because of dehydration. And of course, it is highly infectious. Typhoid fever affects the small intestine, and it is characterized by fever, and abdominal pain, of course, just like gastroenteritis, abdominal pain. But there are two characteristic symptoms in case of typhoid fever. The first one is constipation. All the disease we talked about were characterized by diarrhea, but here it is characterized by constipation and severe headache. But headache is a non-specific symptom because all types of fever cause headache, but here the headache is very severe. Insect-borne infection, we've discussed this before. We have in the first semester, and we have mentioned letter C, which is food poisoning through flies. This is a type of insect-borne infection. You also have malaria, and we talked about malaria several times because malaria is caused by a protozoan, which means that it is a unicellular organism, and that unicellular organism is eukaryotic, means that it has a true nucleus and it circulates in your blood if you are infected. And there is a mosquito, a female mosquito that belongs to species Anopheles, female Anopheles mosquito that bites you with its piercing mouth parts and then sucks a dose of blood if you are infected, and goes to pierce the skin of another person and pour this infected blood and transfer the infection to it. So we have two human hosts. The first one who was originally infected, who was bitten by the mosquito, and then the mosquito, which is the insect vector, will transfer Plasmodium malaria protozoan to another person to start a new life cycle there. The last thing is the bubonic plague. Bubonic plague is put under insect-borne infection because bubonic plague, the insect vector here, the vector here is the the rat flea. The rat flea. And the microbe that is carried by the rat flea, that is transferred by the insect vector rat flea, is Yersinia pestis. So Yersinia pestis is a bacteria. So if you have an area that is full of rats, and of course, you know that the rat is a big mouse, and if there is an area where rats are abundant, then there is a high risk that there might be rat fleas, which are the insects on top of their bodies, and some of these rat fleas might have inside their body Yersinia pestis. If this rat flea bites a human host, then Yersinia pestis will be introduced into that host, and it causes this disease, which is bubonic plague. Why do we call it bubonic? Because it results in buboes or swollen lymph nodes. It results in buboes or swollen lymph nodes. Swollen lymph nodes. And why do we call it plague, or not why actually? What is another symptom of bubonic plague, or a synonym of bubonic plague, is Black Death. Why do we call it Black Death? Because actually, as you can see here, when the foot is affected by bubonic plague, there is a part, the distal, the far part of the foot, which are the toes, become black. And they become black because they become dead. So the whole human body is alive, but the toes consist now or are now consisting of dead tissues. This means that they are gangrenous. Gangrenous. What is gangrenous? Gangrenous is the adjective and gangrene is the name. Gangrene is the death of the tissues and them acquiring this black discoloration because of it being infected with a certain type of bacteria like Yersinia pestis that release toxins that cause death of the tissues. And once they become dead, they do not receive the blood supply and they look like this, they look black in color. And of course, if you don't amputate them surgically, they will be fragmented and they will be detached from the body. But the best thing here, in addition to giving, of course, the patient the specific antibiotic in order to kill Yersinia pestis because it is a bacterium, but unfortunately, the patient will have a foot amputation because here when you amputate, you don't amputate only the gangrenous parts, but you amputate one joint distal to them in order to protect the rest of the body against the spread of the toxin and against death. Thank you so much. This was all what I wanted to deliver to you today, and you will be receiving a relevant sheet to submit, inshallah, on the first day after the spring break. Enjoy your vacation and see you on Sunday, inshallah.